Hi everyone, my name is Zhijie Wu. I'm a research fellow from Hematology Branch, National Heart, Lung, and Blood Institute at the NIH. Today I'm presenting immune cell crosstalk in deficiency of adenosine deaminase 2. You may probably know better than me about this disease. DATA2 was first defined in 2014 as a monogenic disease, and its clinical manifestations now encompasses early onset vasculitis, vasculopathy, recurrent stroke, systemic inflammation, immunodeficiency, and hematologic abnormalities. And its clinical phenotypes is caused by the reduced activity of ADA2 due to autosomal recessive loss of function mutations in ADA2 gene. ADA2 is primarily expressed in monocytes and many other myeloid cells, and ADA2 plays a role in maintaining the balance of macrophages, including M1 macrophages, which is pro-inflammatory and can produce TNF-alpha, and M2 macrophages that are anti-inflammatory. And in DATA2, the decreased ADA2 activities leading to the accumulated adenosine in extracellular microenvironment. This leads to the increased pro-inflammatory cytokines, including TNF-alpha production by M1 macrophages, and then could lead to the neutrophil extracellular traps. And all of these contribute to the inflammation and tissue damage in DATA2. We previously have performed the single-cell RNA sequencing in peripheral blood monocytes in a cohort of 14 patients. And with this analysis, we identified that the non-classical monocytes are overrepresented in this cohort of patients, and instead, the classical monocytes are decreased in these patients. And by analyzing their gene expression, we identified that the immune-related genes are upregulated in different monocyte subtypes, and those genes are highly enriched in interferon signaling and also antigen presentation process. We also observed that interferon gamma and TNF-alpha protein levels are increased in these patients. Well, based on this data and the prior knowledge, we think that due to the mutation of ADA2 gene, adenosine accumulated in extracellular microenvironment, and this lead to the interferon gamma production, which can then promote monocytes differentiation to M1 macrophages. And M1 macrophages can then release TNF-alpha. And all of these contribute to inflammation and tissue damage in DATA2. As much work has been done to study the myeloid cells, including monocytes, neutrophils in DATA2, we found that lymphocytes like T cells and B cells are relatively under-characterized. In the initial report of the DATA2, there are little impairment of T cell compartment. The naive and the memory T cells, the mitogen-induced proliferation of CD4 and CD8 T cells, and also their cytokine production in response to stimulation are in general comparable in the patients compared with healthy controls. And there are a few recent studies that try to profile lymphocytes in DATA2 patients. In a cohort of 9 patients, the authors observed the CD19 positive, CD25 positive B cells, memory T cells, and NK T cells are decreased. And in another cohort of 14 patients, the authors observed the defects in B cell response and T follicular helper cell function. And we noticed that in one cohort, the authors observed the lymphoproliferation in a few patients, And in some of these patients, there are clonal expansion of CD3 positive, CD8 positive, CD57 positive, phenotypically cytotoxic T lymphocytes. And they also have clonal V-beta expression and STAT3 mutation. And the authors also profiled the subsets of CD4 and CD8 T cells, including naive, central memory, and effector memory T cells. However, due to the large normal range and the big variations among samples, they didn't observe a significant difference in terms of those T cell subsets in patients compared with healthy controls. So, as I talked before, we previously performed the single-cell RNA sequencing of monocytes in this cohort of 14 patients. And there are two sets of siblings in these 14 patients. Patient 3, 5, and 6 are siblings, and patients 7 and 8 are siblings. And among those 14 patients, we further performed the single-cell RNA sequencing of their T cells in 10 of these patients. So this is the clinical characteristics of this cohort. There are 5 females and 9 males, and with median age of 23-year-old. And the disease-associated variants spanning the entire ADA2 gene. And among those 14 patients, there are 7 patients, they have hematologic phenotypes, including lymphopenia, neutropenia, thrombocytopenia, and hypocellular bone marrow. And they also have other systematic manifestations, including low immunoglobulin and the skin clearance. And the majority of these patients were treated with TNF-alpha inhibitors. So with coupled single-cell RNA sequencing and the single-cell T-cell receptor sequencing, we were able to profile more than 200,000 CD3 positive T cells from peripheral blood from 10 patients and 5 healthy controls. And we projected the single cells to a 2-dimensional UMAP based on their transcriptome similarities. And we were able to annotate their phenotypes, including CD4, CD8, and also naive, central memory, and effector memory T cells. So in order to resolve the heterogeneity of T cells. So with this analysis, we compared the ratio of CD8 versus CD4 T cells, and also the percentage of different T cell subsets, including naive, central memory, and effector memory CD4 and CD8 T cells. And by doing this comparison, we found that there's no significant alterations of T cell subsets in DATA2 patients. We then analyzed the TCR usage in these 10 patients. And we found that the TCR usage in these 10 patients are, in general, multi-clonal, which means that their TCR, the TCRs in these patients are expressed at the basal level, similar as in healthy controls. And the TCR usage are not shared among patients, which means their TCRs are quite individual private, rather than disease-specific. So this data indicate there is unlikely a common pathogenic leading to the T cell activation. So then we compared gene expression of their T cells with T cells from healthy controls. And we saw the activation of both CD4 and CD8 T cells with upregulation of interferon alpha and interferon gamma pathways. And we identified interferon gamma gene activation as the signature of their T cells, with STAT1 as the hub gene in the T cell activation, in the gene network of T cell activation and the cytotoxicity. So the next question we ask is, those T cell activation we observed here is the initiator or contributor to the disease pathogenesis. So the next analysis that we performed is to check the cell-cell interactions between T cells and monocytes by analyzing the ligand or receptor expressions on T cells and monocytes. So first, I used an algorithm called CellPhoneDB. It profiles the ligands and the receptor interactions between T cells and the monocytes separately in patients and healthy controls. And in general, the cell-cell interaction between T cells and monocytes are distinct in the patients compared with the healthy controls. And then we used another algorithm called NicheNet. With this algorithm, we are able to identify the top ligand receptor interactions and also further their downstream targeted genes. So with this method, we actually found many of the top ligand receptor interactions likely contribute to the upregulation of STAT1 gene. For example, here, we found that the HLA-A HMGB1 ligand from monocytes interact with receptors on T cells, likely contribute to the upregulation of STAT1 in T cells. Similarly here, we identified that TGFB1 PTPRC ligands from T cells, they interact with receptors on monocytes, likely contribute to the upregulation of STAT1 in monocytes. So with this study, we try to define the involvement of T cells and monocytes in the pathogenesis of DATA2. We think that the activation of the interferon JAK-STAT1 pathway remains the key feature of activation of T cells and monocytes in DATA2. But meanwhile, the interaction between T cells and monocytes likely lead to upregulation of STAT1. Thus, this augments the response of cells to interference upon stimulation. So in DATA2, the trigger of the disease still is the mutation of the ADA2 gene, leading to reduced activity of ADA2 enzyme. And then the monocytes skewed to M1 macrophages. But the study here showed that T cells somehow they interact with monocytes by ligand receptors, and these interactions likely augment the inflammation and cell activation. So in summary, in the current cohort 14 patients, there are no significant alterations of T cell subsets, and there was no clonal expansion of T cells. Most TCRs were expressed at basal level in patients and healthy donors. And TCR usage was private to individual patients and not disease-specific. And CD8, CD4 T cells are activated with genes upregulation in interferon pathways and with STAT1 as the hub gene in the gene network of T cell activation and cytotoxicity. T cells in DATA2 patients showed distinct cell-cell interactions with monocytes, and many of these ligand receptors likely drive upregulation of STAT1 in both T cells and other immune cells. Because immune cells or cells, they are never acting alone. They are always crosstalk with each other. So we believe this cell-cell interaction analytical pipeline and could have broader applications. So we also applied the similar approach to VEXAS, which is a disease newly identified by Dr. David Beck and Dr. Kastner's group. VEXAS is featured by autoinflammation disease in adult men due to the mutation of UBE1 gene in the protein ubiquination. We did single-cell RNA sequencing of their bone marrow, hematopoietic, and immune cells, and we were able to identify that the inflammation originated very early in their hematopoietic stem and progenitor cells, and also more obvious in the mutated cells. But what we identified with the cell-cell interaction analysis is that the crosstalk between the mutated cells and unmutated cells, and also between hematopoietic cells and immune cells, likely augment the inflammation in these patients. That's pretty much for today. Thank you.